Okay, so finally we're getting to this. Before we begin, I want to just state a few things. First of all, if I mean, this is for myself, so I need to state a few things for myself, and it will come to help, I think, other people as well if they come to listen. Um, this these notes, they do not refer to everything that's needed in the biology syllabus. Um, you know, there are certain topics within topics. So, some very certain specializations going down. Um, those are not really mentioned in this document, from what I've seen. Um, so you need to keep that in mind, um, because it can get rough, especially when you're trying to study the entirety of the syllabus, and there seems to be some certain few things which are missing. It can get rough when those things are not there. So yeah, this is just to state that this is not everything. This is, I would say, the most basic stuff, the most important stuff. And as you go down, the fundamentals, and you get more specialized, um, it's just not gonna be there. That's something that you have to go in on your own. I have certain books, the textbook, the specific IAL textbook for biology has everything you need. Um, and if you, for some reason, want to go further than that, then there are other textbooks. I myself have, Campbell Biology, which I have never gone through. I don't know if I'm allowed to open this on YouTube, but I don't really care. So we're gonna do it. Um, if the video gets taken down, well, let's do that. There's a lot of stuff on here, but we're gonna start off with the most preliminary of things. That's why we're not gonna go into Campbell Biology just yet. I think when we start going into stuff more specialized, like if you look into the specification and look into stuff that's more specialized, take this for example, or take this process, this role over here, for example, or we talk about fertilization, role of mitosis. These things are they can have a lot of different interpretations, and I think it's worth checking out Campbell Biology's version of it. So you can always do that, and we also have ChatGPT here in case whenever we need to do that. So we'll do that. I don't really know why I spent 3 minutes explaining myself. That's not really necessary. Can you see ChatGPT? Well, wait, can you see everything? I'm not sure what's going on here. Oh, okay, you can. Oh, I see this. Wait. Oh, I see what's going on. Okay, this is funny. Okay. So then, no, I don't want to move my display capture. Wait, what was that? Are we live? Okay, I'm not sure if I was recording during that entire thing because they don't really make it very clear. Okay, hopefully this is a bit nicer to look at. If I do this, has anything changed on stream? I don't think so. This is just really neat to me, so I'm not sure. Actually, one sec. Let's begin. Actually, before I begin, let me just set up my desk because this shit is messy. Okay, and let's increase the volume. Okay. So we're starting off really, really, really basic. Okay, we're going to talk about cells. Read Song of the Cell by Siddharthi Mukherjee or something like that. Pretty good book. No. Okay, so cells. Before we start, let me just, can we get a little bit bigger? I feel like this is a bit small. Why are we in inches? What's going on here? Okay, yeah, that's fine. What is, okay. Okay, so again, we still have time. It's already been about 5 minutes into the video. That's okay. So we're going to be looking into these things. Cell structure first, then we're going to talk about reproduction, and then lastly we're going to talk about development. Reproduction, as you can kind of, I hope you can kind of tell, it's going to be talking about mitosis. Oh well, it's going to be talking about meiosis first, and then it's going to be talking about mitosis, and it's going to link mitosis with cell development, stem cells, differentiation, all that business. So when we start off, we're talking about, wow, that's loud. Sorry about that. We're going to be talking about the divisions. So we're going to have two types of cells. We're going to have eukaryotic and prokaryotic cells. And we're going to give some examples. So bacterial cells are prokaryotic cells, where human cells are eukaryotic. This is the biggest or most apparent differentiation between the two. Eukaryotic cells contain a nucleus and a membrane-bound organelles, where prokaryotes don't. That's the biggest difference. That's difference number one. Eukaryotes, again, contain nucleus and membrane-bound organelles, and prokaryote cells don't. This is the most primary kind of difference. Okay. So remember, we are human. We have human cells. We have eukaryotic cells. We have nuclei, and we have membrane-bound organelles. Membrane-bound organelles being inside your cell, inside the cell's membrane, we have organelles, stuff in there, which function for the cell of the main activity of the cell. They do the stuff, and these are present in our cells, where it's not present in other people's cells, in bacterial cells. Okay. Most eukaryotic cells have the same types, same internal organelles, but cell specializations mean cells often differ in number and sometimes types of organelle present. So, for instance, red blood cells have no nucleus, so they have more room for transporting oxygen. Yeah. So, basically, we just mentioned that they need to have, like, our human cells have nuclei within them. However, straight away we have like a little contradiction here. Red blood cells, which are obviously, present in our human body, and so are eukaryotic cells, they have no nuclei. So are they prokaryotic? Wrong. No, they're not. They are nuclei. They are eukaryotic. But they're a product of cell specialization. So although they can have mostly the same internal organelles, different specialization will lead to them having sometimes different number of organelles, and sometimes even different types of organelles present. Okay. When does cell specialization occur? When your body's in harsher or different environments, and they need to come up with stuff to they need to adapt to house different, like, so that they can function at optimum efficiency in those different environments. That's why they specialize. Take if I was like a snake, right? But then I suddenly get sent to like a really cold environment. So now my scales and stuff, they don't really help me anymore. So I now need to start conserving for like heat. Like I need to start conserving my heat rather than losing it so much, because that's what snakes are kind of like used to doing in the desert. So now they might start growing fur and shit. That's not really there in snakes. But if it does start growing fur, does that mean it's no longer a snake? No, it is a snake. It's just specialized because now it's in the different colder environment. Okay. Fur because they want to, because it decreases heat loss or whatever. So now how are cells organized? Because now we've talked about how cells are categorized and how cells can differ although they are categorized, like why conflictions can occur although we categorize them. But now, if we further want to organize these cells, right, especially in our human body, this is going to be talking about mainly our human body. I want you to visualize this in the human body specifically. Okay, you can't do this for any other, but let's do this for the human. So, if we start from the bottom, right, we have cells, and we work our way with groups of cells that carry out a common function in form of a tissue. So, if you have a group of cells, then they work together as a tissue. And if you have a group of tissues that work together for a common function, then they work as an organ. And if you have a group of organs, that's an organ system. And if you have an organ system, then you practically have an organism. Okay? Can't have an organism without an organ system, unless you're unicellular or something that's very small that doesn't require it. Okay, so organs and organ system. Okay, pretty easy to understand, but this is actually a very important point. It helps you understand, well, this necessarily doesn't this doesn't necessarily help you in the sense of the great scale of things. However, if you can understand this, the fact that an organism, a larger organism, needs tissues, needs cells, needs tissues, needs organs, and needs organ systems, then it can kind of help you to understand how it can differ depending on which organism. Because some organisms don't have organ systems, some organisms don't have organs, some only have tissues, some only have cells. So yeah. Now, if you have an example here, groups of cells in the stomach make up this muscular tissue, and the tissue alongside the epithelial tissue make up the tissue or the stomach organ, and the stomach and various other organs such as the pancreas and the small intestines make up the digestive system. Then many organs are part of more than one organ system. So you can have the pancreas, which is both a part of the digestive system and also the endocrine system, because it releases hormones, I think. Now, how can we see more detailed pictures of cells? We just use we find the okay. I don't know why he wrote ultrasound of cells. That's wrong. That's hilarious, actually. Though. Alright, that's correct. I don't know, but you can find the ultrastructure, which is the more detailed structure of the cell, using a microscope. You got the light or the electron. We'll learn about those later. Okay. This picture over here, you won't be able to see it with a light one, only an electron microscope. So if you were ever at school and you're trying to look at a cell, look at your little cheek cells or whatever, and you can't see this sick picture, that's probably because your microscope that you're using is very tiny and it's very weak and it doesn't have high enough magnification in order to see the subcellular organisms. The membrane-bound organelles, these things. Okay, so just talking about a little bit about the ultrastructure. We have a very common one. We're talking about the nucleus, and we start off with the nucleus talking about how it's surrounded by a double membrane. So immediately this kind of can give you a sense of sorry about that. So it can give you a sense of, where else have you heard about a membrane? You've heard about a cell having a membrane. So a nucleus is separated by a double membrane. What does it mean by a double membrane? Well, it's literally just it's literally one membrane over here alongside another membrane over here. You got two membranes. Why is that? Perhaps extra protection, whatever. So now, that's maybe something that you can look into. We can ask ChatGPT that later, maybe. So it can contain pores, which then allow molecules to enter and leave the nucleus. So here is the properties and function. So we have one property over here, right? Call this P. Think of Pro. So the property is a double membrane, which contains pores. And the function of this is we have it's allowing or enabling molecules to enter and leave the nucleus. Now, if you've studied in paper 1, those can be, the things that are leaving the nucleus could be your mRNA, right? Am I remembering that correctly? I'm not sure. Your mRNA that's leaving to meet the ribosomes to turn to to form the proteins, right? So that's going to be your mRNA. Anything else that could be leaving? Well, perhaps ribosomes. It's sorry. This is I'm doing this in the wrong area because I was thinking this entire thing is the nucleus. No, it's going to be leaving through these tiny holes over here, right? So it's leaving. Your mRNA is going to be leaving from there, okay? Like through there. So yeah, the nucleus also contains chromatin and nucleolus, which is the site, and nucleolus, which is the site of ribosome production. Chromatin is going to be your stuff over here, and your nucleolus is going to be your center thing over here, denoted by this. And it's giving you the function already. Ribosome production is the function of the nucleolus. That's what's going to be happening mainly. As you know, well, we don't know how ribosome production, the process of that, but we do know how proteins are formed, so perhaps that has something to do with it, okay? Now, this is a new thing. You have your rough endoplasmic reticulum and also your smooth endoplasmic reticulum. So you have a series of flattened sacs enclosed by a membrane with the ribosomes on the surface. So, okay, this is a bit weird to say, but as long as you have the picture, I think it's fine. So we have the rough endoplasmic reticulum being shown over here, okay, and you can see the smooth endoplasmic reticulum right over here, okay, and you're gonna see that they do look a bit different. The Golgi apparatus looks more similar to the rough endoplasmic reticulum, but the point being is that these are subrelated. Okay, they are related, but you can see these kind of look like double membranes. Okay, but in fact they are flattened sacs. So they are like sacs. Imagine this big, but flattened to be this big. Okay, and after you flatten them, they are enclosed by a membrane. So they are enclosed. Truly, they are enclosed or surrounded by a membrane, which is studded or kind of repeatedly stuck, over here, repeatedly stuck, as you can see those little dots, with ribosomes on the surface. So not only is it enclosed by the cell membrane, but it's also studded with loads of ribosomes as well. The RER folds and processes proteins, so this is going to be your function over here. You're given your little, so it folds for a greater surface area, perhaps it's just so that it can fit more ribosomes and more endoplasmic reticulum in a smaller space, and it processes proteins made on the ribosomes. That's kind of just seems like common sense. When you have ribosomes, you're going to have processing proteins. Now, your next bit is going to be your smooth endoplasmic reticulum, something that's kind of subrelated to the rough endoplasmic reticulum, and it's going to be a system of membrane-bound sacs. Okay, so again, we're talking about the membrane sacs, or like the sacs enclosed in membrane. It's the same thing. However, it produces and processes lipids, okay, and carbohydrates, I believe. But again, this is just this is what I mean by interpretations. Okay, over here it says lipids only, but if I go over here, and spend some time looking through that, we'll probably see something different. So that the point is that you kind of just get the image of what it does, and then you can specialize. You can look into the specialities of what it produces or what its function is later on your own free time. Okay. So It's a system, the smooth endoplasmic reticulum. It is a system of membrane-bound sacs. So they are membrane-bound, as in they are enclosed in a membrane, which is enclosed in the larger cell surface membrane, and it produces and processes lipids. So lipids are pretty important, and you just need to know that they are produced in the smooth endoplasmic reticulum. I think that's the greatest important factor that you need to know over here. So you just need to know lipids made here. Okay, you learned all about lipids in unit 1, so now you know where they're made, not how they're made. Well, you know how they're made, but not specifically. Yeah, they're made here. Alright, I'm back. So let's continue. So we learned about where the lipid production is taking place, and that's going to be usually in your smooth endoplasmic reticulum. And if we moved on, now we're talking about the next new thing, the Golgi apparatus. Okay, so this is your series of fluid-filled, flattened curved sacs with vesicle, blah 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 blah. A lot of garbage. Let's first look at it. So first of all, you're gonna notice that it's kind of got a similar structure to the smooth endoplasmic reticulum and the rough endoplasmic reticulum, mostly the rough endoplasmic reticulum. Only difference being it's not studded anywhere, and it's got these loads of like bulges, and coming out of the bulges you have like these bulges which aren't connected, and these are called vesicles. Okay, and you can kind of see the vesicles contain stuff, and at one point vesicles can be used for secretion. Okay. So now that we've kind of like visualized it, we can see that there are a series of fluid filled. So the stuff inside were fluid, flattened, which we saw, kind of flattened just like the sacs in the previous ones, flattened and curved sacs. Okay, why are they curved? Because if you look at it, I mean, they've kind of got that curve. Over here, these are curved, but they're only curved because they're around the nucleus, and these aren't even curved at all. But these are curved. They have a long side, they have a short side. Okay. Now, they're flattened, they're curved. Flattened is the usual. You can kind of imagine when you see sacs, then you can kind of imagine they're flattened, especially when it comes to membrane-bound organelles, and it's fluid filled. This will come into this property. This one specific property out of all of these properties, this one specific property will be explained later. And the fact that they're curved, this is a special one. This is a special one to Golgi apparatus. Okay. And these sacs, there are a series of them with vesicles surrounding the edges. Okay, so you're gonna have your sacs, and this is gonna be your special property as well. It has vesicles around the sacs, okay, surrounding them. And what the Golgi apparatus does, okay, its function to say, it processes and packages proteins and lipids, and it produces lysosomes. Okay. Notice how we don't mention really anything regarding vesicles from here to here. Not sure why that is. I thought there would be something related to that, but if there isn't, then there isn't, and that's my bad. So yeah, it is surrounded by vesicles, but when it comes to actual function, it processes and packages proteins and lipids into vesicles. If I'm not wrong, I might be wrong about that. I'm pretty sure it packages stuff into vesicles so that they can be transported around. Okay, and it also produces lysosomes, which we'll learn about later. Yep. Okay. Furthermore, mitochondria. This is the good old powerhouse of the cell type shit. Usually oval shaped, bound by a double membrane called the envelope. So first property, oval shaped. Second property, double membrane, as you can expect. Envelope, similar to the nucleus. This is actually an important similarity to make. The fact that it's close to the nucleus, it's like similar in structure to the nucleus. Each membrane has a different function, but the only one that we need to really care about is the inner membrane, because that one is folded to form projections called the cristae. So it's folded to create special structures called projections, called cristae, more specifically. And it produces also a matrix, which are basically the gaps in between on the inside, which contain the enzymes that are needed for respiration. Okay, cellular aerobic respiration, rather aerobic. So if you look at the mitochondria, you're gonna have the outer membrane over here, and the inner membrane, the yellow one, over here. And the inner membrane is folded into a cristae with little gaps. It's folded into it's just folded. And it forms projections called cristae with a matrix inside. Okay, well, it does fold to form a cristae. So this structure, you could call it a cristae, like folding shape, with these little gaps in the middle, which I'll highlight in red, right? These little gaps, if you can see them, these are going to be your matrix, where it's fluid filled, and it also contains the enzymes needed for respiration. Moving on, and this is obviously aerobic respiration, using ATP, or rather making ATP through the Krebs cycle, which we'll learn in a later unit. So, centrioles next. This is also new. Ribosomes, not so new. Lysosomes, also new. Centrioles, they are hollow cylinders which contain a ring of microtubules. So, first of all, we're already having two things. You can think of centriole as a center, right? It kind of gives that vibe. And if it has center, then you can kind of think of it as a circle. And if it's a circle, that means it's a ring, correct? And if it's a ring made up of microtubules, then you can kind of imagine that those microtubules, you can imagine if it's a ring containing smaller tubes, then the main thing is just going to be a big hollow cylinder or a big hollow tube. So you can kind of think of it as a big hollow cylinder containing a ring of microtubules or microtubes or micro cylinders arranged at 90 degrees or arranged at right angles to each other. Okay, so, and this is kind of hard to visualize even for me, so if we went to the image and we looked at a centriole, you can see that we have two. We have one on the bottom and one on the top, and these are at a 90-degree angle. Why is that at a 90-degree angle? Well, that's actually pretty interesting. You can figure that out on your own. I think that's a pretty cool thing to look up. Centrioles are involved in cell division. This is actually going to be something that we're going to be referring to later in this video when we talk about mitosis, and that's the spreading apart. And the spindle fibers, those are actually involved in the centrioles. Okay. Ribosomes, next up. These are composed of two subunits, as you know. So again, they don't mention these subunits here. This is something that would be mentioned in the textbook, but isn't mentioned here. This is also why I think it is useful to look into the other textbooks, or look into these school textbooks for further understanding. But honestly, let's okay, I might have made a blunder here. Hold on. Alright, it's been a while and my computer kind of exploded, so ignoring what just happened. I don't really remember what we were talking about. Ribosomes, two subunits which we didn't really discuss. I think it was like 50S and like 30S or something like that. Starter protein production, you knew this already if you did Unit 1. Unit 1's, what's it called? Protein synthesis, right? Lysosome, it's a vesicle. So this is finally where we're using the term vesicle, which contains digestive enzymes, so it breaks stuff from complex to simple so that it can be digested. So it contains these digestive enzymes and it's bound by a single membrane. If you do know this, this membrane can be broken and so all the digestive enzymes are going to like leak out everywhere and just destroy everything and killing the entire cell. Now, that might sound really bad, but if the cell wants to die or needs to die through programmed cell death, apoptosis or whatever, it will do that. Pretty cool. But in general, this vesicle or this lysosome vesicle is used towards breaking stuff down, waste stuff, or etc. etc. You can just kind of imagine. It's not that hard to visualize. Now, this was your eukaryotic stuff, and that's why we included, all these cell membrane as membrane-bound organelles and the nuclei. But now we're talking about prokaryotic cells. We're going to be talking about, none of that stuff. We're going to be talking about, well, you might think, if it's membrane bound, if there's no membrane bound inside a prokaryote, then what is all this? Well, that's the thing. Ribosomes and genetic material are not, in fact, they are not in fact considered membrane bound organelles. Well, I thought the ribosomes were, but I guess not. Okay, but anyways. So we're beginning off with a cell wall, which is present in prokaryotes, okay, not in eukaryotes, other than plant cells. In animals, you're not gonna find them. Just a rigid outer covering made of peptidoglycan or whatever, and in plant cells, those are going to be made out of cellulose. Just want to remember that. Capsule, which is going to be inside or like beneath or like under the cell wall. It's going to help you retain moisture and adhere to surfaces. So these are the two functions of that. Adhere would mean stick to other surfaces, and to retain moisture is just when it's in a dry environment to survive. Plasmid, circular piece of extra DNA. So there's just extra genetic material, which it might contain some some genetic material that can help it survive. So maybe perhaps it'll contain genetic material for let's say for the MRSA, I think it was called, bacteria, the ones which are extra resistant to antibiotics, right? Those extra DNA, that extra the genetic code which codes for that extra resistance to bacteria would be in this plasmid. Now your flagellum, is used for transportation, tail-like structure. Villi, hair-like structures which attach it to other bacterial cells. I also hear that this is used for, disguising themselves as, because disguising themselves as other cells, or as not as pathogens, and you would want this because your surface cell antigens is what gives it away, right? So if you have Pili, perhaps you can go undiscovered, and it prevents death from phagocytosis from a white blood cell or by the immune system of whatever organism the bacteria is inside of. Ribosome, protein synthesis, makes sense. Mesosomes, this is probably the only new one. I hope you remember or most of these except for let's say the pili and the capsule. So what the mesosomes is infoldings of this inner membrane. So you can kind of imagine the cristae inside the inner membrane of the mitochondria. So there's going to be infoldings. It's not present in the image, but infoldings of the membrane, which contains enzymes required for respiration. So it really truly is like the mitochondria's, cristae and matrix, the mesosomes. So lastly, we're going to be talking about enzymes, extracellular. There's also intracellular, but I suppose only extracellular we're going to be talking about here. Secreted by cells into the external environment, cytoplasm, whatever. Enzymes are, well, sorry, not the cytoplasm, the solution, or perhaps if it's a cell inside a cell or something like that, then the cytoplasm. Enzymes are proteins, right? So they're synthesized in ribosomes, right? Because that's where protein synthesis happens. Free in the cytoplasm are attached to the rough endoplasmic reticulum. As you remember, they could be free floating, or they could be, studded in the RER. Polypeptide gets sent into the Golgi apparatus where it's folded into its 3D shape. Then it's packaged as a Golgi vesicle. So first of all, Golgi apparatus. What do we remember for the Golgi apparatus? Well, we do know that it packages them, right? That's what we remember. But it also processes. So it processes and packages proteins and lipids. So remember the first two. So there's two parts. Okay, so we can kind of we should put these into two parts. So it processes and it packages proteins and lipids. Okay, so in this case, we can imagine this as after the protein or the polypeptide is made, it then gets sent to the Golgi apparatus where it's processed in a way where it's folded and into its 3D shape. Then it's packaged into the Golgi vesicle, where then it can fuse with the cell surface membrane to release the enzymes outside of the cell. As you remember, that's the vesicle, that's the cell surface membrane, that's the vesicle, it goes over there. The membrane then fuses, and the inner stuff, which contains your proteins, hopefully, gets secreted outside into the external environment as extracellular enzymes. Now, these vesicles are important to ensuring, so the function, why are we using vesicles? Well, vesicles are important to ensuring that enzymes only catalyze reactions, such as the breakdown of biological molecules were required, in this case outside the cell. So it's not really, okay, that was a bad statement. It's not really that the vesicles are an important part, but why are we using vesicles, and why are we, secreting them outside? So that enzymes can catalyze reactions, like the breakdown or etc. And in mostly, because mostly they'll happen outside of the cell, so you'll need something to take them out. That's why you need the vesicles. Okay. Now we're going to go through these. Oh man, I hate this so much. All right, let's get back into this. So studying cells, talking about the basics of how we view cells, how we view the membrane bound organelles. How do we see these things? How do we know how these things function and how they look, right? So to understand the function, then we need to be able to look inside and study the cells, right? Makes sense. If you want to understand how a car works, you want to look at the engine, you want to look at what's inside the engine, what fuel you're using, etc., etc. And the study of cells, looking inside using a microscope, there's two factors. There's the resolution, the magnification power, and the ease of use. Three factors, my fault. Resolution, definition, important. Degree to which it is possible to distinguish between 2 points that are close together. You have this point, you have this point. Am I able to distinguish them between x length, or am I only be able to distinguish them with x minus 7 length? So the closer they get, at what distance will I actually be able to distinguish them? I hope that's understandable. If, let's say, they're this close and I'm able to distinguish them, but the moment they get a bit farther, it would be more like if I have 2 points like this and this, okay? But since the resolution of my eyepiece is bad, they will look like they're the same thing. They'll just be one big thing. However, if my resolution was greater, then I would actually be able to see that small distance in between them. That is basically all you need to know. So, the degree to which it is possible to distinguish between 2 points that are close together. That's going to be your resolution definition. It's important. You should try to remember this. Magnification now. Degree to which an image is larger than the object itself. The image of an object is larger than the object itself. Okay, this is going to be your second definition. Pretty important. Ease of use is just, portability. Maybe like, the price, size leads with portability, leads with price as well. These kind of things, or like, how easy it is to use. Like it is to use. That just generally, you'll see it when you'll understand it when you see it. You can understand, or you can calculate image size by using the formula. This is by this. I don't need to stick on this more. I really think this chapter is a waste of time. Optical microscopes, you'll have used in school. You can barely make out anything except the nucleus. So electron microscopes are used more in actual labs. Two types: there's your SEMs and your TEMs. Scanning electron microscope and transmission electron microscope. And we'll go on talking about these now. So, your optical microscopes, all right? This is going to be your light microscopes, right? Your light or your optical microscopes, the cheap ones, right? So they're easy to use, your main point. They're easy to prepare, and you can use live specimens, because all you're doing is that you're using light to form the image. You're not shooting any electrons, you're doing nothing to the actual subject. You can even look at live specimens, so live creatures. You can see living organisms in the thing. You can see them moving around. So if you're looking at a picture, and there's some microorganisms that are moving around which are alive, bacteria and such, you can tell that this is a light microscope footage that you're seeing. Light has a longer wavelength than electrons, so the microscope has a lower resolution. Okay, so this is going to be your disadvantage. Lower resolution. Why? Because light has a larger wavelength. If you remember, when we were talking about in physics in my previous video, when we had a larger wavelength, and that was going towards the scanning image. Since the larger wavelength covers a larger area, you can't, like, the small minute changes in the surface cannot be scanned. Something like that. I don't remember the same exact words. I could open it right now. Let's see, because this is actually kind of interesting. Is it on here? Then I'll have to look at the video itself. There you go. As the wavelength of the light increases, of the waves increase, the less fine details can be resolved, meaning the amount of information that you could obtain decreases. The same thing is happening here, in the sense that, when light has a greater wavelength, you're picking up less fine and minute details, so the resolution will be lower, okay, as the wavelength increases. Okay, next, lowest maximum magnification. Why? Because well, generally it's light. It's bigger than electrons, much bigger. So it's made out of photons rather than electrons. So it's going to be, you're not going to be able to see that small of an image. So the magnification will be smaller, and so since the magnification will be lower, your large organelles are going to only be visible. No small organelles. Okay. Scanning electron microscope, and so you have your SEMs and your TEMs, right? So let's go into those. Scanning electron microscope, remember, it scans, and this is transmission. If you remember from physics, again, transmission is when you go into it, and the reflection was where you just bounce off, right? So there's your reflection and your transmission, let's say. Okay, so scanning scans a beam of electrons across a sample which knocks off electrons and forms off an image. So you have your beam of electrons which knocks off electrons, which forms an image. Okay, so yeah, okay, knocks off electrons, which forms an image. So you're gonna have like the surface, and it's gonna knock off electrons, and then it'll form the image as it comes back up. So you're gonna have higher resolution than optical microscopes, of course, higher magnification than optical microscopes, of course. Forms 3D images because you're bouncing off and you're hitting it. So different surfaces, different ways it's gonna bounce off, helping to form a 3D image, and can be used on thick specimens because it's not going to pass through, so it's going to only hit, so you can have thicker specimens and it'll still work. Lower resolution than TEMs because, of course, if you're going into it, then you can see the more minute details, so it'll be a lower resolution because we're only seeing the surface on the SEMs. You're only seeing the surface, so the resolution will be lower. So TEMs pass electrons through a thin specimen, hence the word transmission. Denser regions absorb electrons, and so less pass through, creating a darker area on the image, right? What this means is that this is let's make this straight. So let's say you have your thin specimen in here, okay? So you shoot the electrons through, and when it goes through and it hits the screen, you can see the image forming, okay? Now, parts which are thicker or denser will absorb electrons. So if I shot electrons everywhere, okay, and there are areas where electrons are passing through, and areas where less electrons are passing through, 1 2 1 2, for example, this would give me an idea that denser regions absorb electrons, so less pass through, creating a darker area on the image. So let's say these parts would be darker because there's less electrons passing through here, okay? So you can then see on the image areas of dark, areas of light, areas of dark, areas of light, which kind of give you a structure of the thing, of the specimen that you're scanning. Highest resolution, highest magnification, of course. Internal structures, because it passes through, and however, it needs very thin specimens because it's passing through, and the slides are hard to prepare because they're very thin. Okay, easy. Staining in microscopes, this is important, and also there's going to be extra information here, which you're only going to be able to find in, for me, I'm going to probably be looking at the textbook for this, because they talk specifically about the different types of, examples of molecules or whatever substances used to stain specific kinds of substances. So it's very specific. Stains and dyes are used to apply to tissue samples. So we're learning specifically stains and dyes for tissue samples. And what they do and how they do it is basically they bind to the organelles, to the small organelles within the nucleus, within the cytoplasm, or within the membrane. They'll bind to them and make them easier to view. Staining increases the contrast of the image formed, so it's easier to distinguish them between two objects that are close together, so it increases resolution. For example, if you have two objects and we stained one of them, or like the stain that we used binds to one of the organelles. Therefore, if it was really close together and through our view, it would appear as only one thing. However, if we stain them, then it would start to appear like this is one bright thing and this is one dark thing, and now we can distinguish these two parts together, although they are extremely close. Okay, so it increases resolution. So as we know, light microscopes are pretty low resolution, it's low res. However, using staining, it increases the resolution. That's the point of staining. Okay, now we're going to reproduction. This is definitely a more interesting topic, so I'm going to pause the video here, and we'll start a new part.